Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com Synthego and BrainXell webinar titled Advancing Disease Modeling for Drug Discovery by Modulating iPSCs with CRISPR and Neuronal Differentiation. I'm Liam Sanyo from the events team here at Scientist.com, and I'll be the host of the webinar. Today, we're being joined by Travis Hardcastle, Senior Product Manager of Engineered Cells at Synthego, and Seth Hansen, who's the Business Development Manager at BrainXell. And together, they're going to discuss applications of CRISPR and induced pluripotent stem cells in disease modeling and drug discovery. And with that, I'm very pleased to welcome the first speaker, Travis Hardcastle. So Travis, thanks so much for joining us today. And the floor is yours whenever you're ready.
Thanks so much for the introduction, Liam. Appreciate that. Thanks everyone for joining us today on what I feel is an important topic, and I'm sure the BrainXell team would agree with me on that as well, which is using CRISPR and neuronal differentiation for advancing disease modeling, particularly as it's used for drug discovery. I'd be a bit remiss if I didn't take a short amount of time to talk about Synthego, where we got started, where we came from, and where we're going. We got our start back in 2016 when Synthego released the world's first mass-produced 100 nucleotide synthetic modified single-guide RNA. This is really our push into the market to create reagents at scale for researchers who wanted to perform CRISPR-Cas9 gene editing in their own hands in their own lab. It was a few years later, in 2018, that we really went to market with an optimized solution for this, which for knockout particularly, which is when we developed our multi-guide strategy for creating knockouts in an algorithmic, creating designs in an algorithmic way to create fragments targeting early exons and create highly efficient knockouts and really optimize CRISPR at scale with that solution. While at the same time in 2018 and into the early parts of 2019, we decided to take these same reagents that we were mass producing internally and start to engineer cells in our own hands, and really went to market with our first, high throughput, fully automated CRISPR cell line engineering platform to really introduce cell models at scale for researchers who wanted to study both knockouts, knock-ins at a high throughput manner. Now in 2020 and into the future, we're really focusing on our GMP capabilities. We just opened our brand new GMP facility just a few months ago out in California, and really focused on being able to create reagents at scale, GMP quality reagents at scale, and really drive researchers closer to the clinic with these guides that we're able to produce in a full GMP format. So what makes Synthego different from any of the other competitors out there, any other providers out there who perform cell line engineering? And it's really the engineering approach that we take to biology with our platform. And really what that means is we design an experiment, we design our automated platforms, we design the things that we do, we build those platforms, we build those automations, and then we test the outcomes of that. And we use those testings to learn from the improvements that we can make with our platforms, from our automated systems to our guide designs. And we take those learnings and we feed those back in to create even better designs, even better, more highly automated platforms, or even better CRISPR designs. And this is applied throughout the whole realm of the Synthego ecosystem, from our hardware to our bioinformatics to our chemistry to our biology and our automated platforms. So Synthego is proud of a few things, particularly the numbers that are on the screen there. If you take at heart the number of gene edits that we've been able to produce from our engineered cells portfolio, it's well over 250,000 edits at this standpoint. When you look at our editing efficiencies, particularly for our pools, we're averaging well over 80% at this point, closer to, with our multi-guide technology, with our newly released express pools, closer to 95, 98% average editing efficiencies for our pools, which is really a great option for our customers. We're really excited about the number of publications that we've been published in. This says over 1,000. I think it's closer to 1,200, 1,300 publications that researchers who are citing and using our CRISPR-Cas9 reagents for their own research purposes. And when you look at where these researchers are based, we ship to well over 45 countries at this point. So really taking a global approach and really getting our reagents in the hands of researchers all across the globe at this point. This can be seen with many of our top collaborators, from St. Jude's to CRISPR Therapeutics and Citro. Many of the top pharma researchers out there are using our reagents at this point to really create cutting-edge research with what they're doing with our reagents. So how does this relate to what we're here to talk about today, which is our CRISPR-Cas9 engineered cells platform? So as I said earlier, we like to take this approach of taking engineering to meet biology and create solutions that really streamline the engineered cells portfolio or engineered cells pipeline. And this is what we typically saw several years ago when you go out into the lab. This is what you'd expect to see: researchers who are sitting down at their lab benches doing guide designs by hand, using informatic tools, taking hours upon hours to do guide design, manually using pipettes to pipette reagents in and out of the wells, or even spending extraordinary amounts of times moving plates around, taking images of their cells or viewing cells as they grow out. So we viewed this and said, where can we create some synergies? Where can we streamline a lot of these processes and make it an expedited process? And that's where we ended up here today, where we are at Synthego. And this is a bit of a peek behind the curtain of our engineered cells facility, which is based in Redwood City, California. And some of the highly automated sort of work cells that we have within our lab space. The image on the right there is something that I wish I would have had back in the days when I was in grad school and when I was doing my early academic research as I came out of the lab. But it's really what I like to creatively call our cell nursery. It's where cells are, particularly for clonal projects, after we single cell print them into single wells on a plate, we allow them to outgrowth, and those plates get moved around via robotic arms. They get imaged every 2 to 3 days. And then within that work cell, we also have machine learning software that tells our scientists, alerts our scientists when these wells are becoming confluent, so that they can manually QC that cell does need to be hit picked and moved into a larger flask to continue the outgrowth phase. And so, as you can imagine, and as we all know, this is the longest phase within a lot of the engineered cells steps of creating engineered cells. And so we reduce the amount of touch points that scientists or that our scientists would have to take into manually looking at how the cells are growing. So what does this all mean for our platform? It really has allowed us to optimize the ability to work with just about any cell line and really look at a 200-point optimization for creating the best conditions for creating highly effective edits. This can be seen with the diagram on the screen from a plethora of different types of cells, from an easy cell type all the way down to a more challenging cell type, as any researcher might know who works with THP1s. These are extremely challenging cell lines to work with. But because of our highly automated platform, we've been able to find conditions to work with just about any of these cells. And as it shows with the cell on the left, a relatively easy cell line to work with, the EMT6, many conditions work really well. And so creating a highly effective editing edited cell is relatively easy to find. But as you start to work towards a more moderately difficult cell line to work with, such as the 5,6,3,7s, not every transfection condition works equally well. And because of this highly elevated platform, we've been able to go in and pick out the condition that works best. And as you can see on the right, some transfection conditions don't work at all when you look at THP1 cells, and they're extremely difficult to work with. But because of this highly automated platform, we've been able to go in, identify the best conditions to work with, and really create highly effective edits with the cell line now in many different types of projects. Some of the things that we talked about earlier, we've been able to now create well over 250,000 edits on our platform. And because of those optimized conditions, work with 800 plus different types of cells. And those are spread out across many, many different tissue types, whether it's breast, prostate, skin, lung, immune cells, brain cells, as Seth will probably talk about later today, colon cells, blood cells. So we've been able to really kind of optimize our editing in any cell types that our customers want to work with at this point because of this highly automated platform that we've been able to generate in California. So how does it relate to some of the topics that we're going to talk about today? It's really because of this highly automated platform that we're able to create really great iPSC cell lines. And here's an example of some of the products that we're able to offer from knockout and iPS cells, from knockouts to single nucleotide variants or SNPs, to even small and larger tags. And we offer this in a plethora of different options. We can use either a homozygous or heterozygous editing strategy to create this, and we can even provide these in the shape of clones or pool formats for our customers, depending on the type of research that they're looking to do. Looking at a bit of how this works on the Synthego workflow and how we look at project from as it moves through the different forms of the platform. This really starts with the project submission. This is really when customers come to us, tells us the types of edits that they want to do, tells us the cell line they want to work with, the gene they want to work with, and the type of modification they want to work, whether it's a knockout or knock-in, or even inserting a tag within that project or within that cell line. Once we know all this information and we're ready to go, we submit that project and it kicks off the process. From there, we do have a highly automated machine learning platform that helps us design our guides. But these guides are also double checked as a bit of a checks and balance with a scientist to make sure that those guides truly are the best designed guide for the particular edit that the researcher is trying to do. From there, it kicks off guide synthesis, and one quick easy step there, because we do have a highly high quality guide. We started off originally by creating high quality guides. This is all done internally with our Halo platform, in which we create synthetically modified synthetic guide RNAs. Unlike some of the different companies out there who are in the cell line engineering space, who use plasmids, we use all synthetic guide RNAs with all of our edits, using an RNP approach with all of our electroporations. From there, after we get the guides back, we start the cell qualification perspective. This is an important aspect to us, as we know that oftentimes the quality of the cell that we put in our platform directly relates to the quality of the cell that we get on the back end of our platform. So at this stage, if a customer has provided us a cell line that they want to work with from their particular lab, we do require that goes through some mycoplasma and IDEX testing to verify the sterility of that cell line before we introduce that into our platform and want to infect any other cells that are on that. We also offer options to our customers at this point to have KRAS hyperpotency testing done on the cells. And we will do this both on the front end and the back end of the process, because we want to ensure customers that whenever they provide us with their cell lines, they're going to get the exact same cell line on the back end from a quality standpoint. So at this point, after it's passed all of those QC metrics, we will begin the expansion process to expand the cells into the proper number of cells to perform the gene editing experiment. From there, it goes right into electroporation. As I said, we electroporate an RNP complex of synthetic guide RNA with Cas9 protein. We do try to target at least 3 different guides per project to move forward with. And so this will be every project, every gene that we edit, we take 3 guides and move forward with the best guide to go forward, and pick the best guide to go forward with. This is really picked up after the electroporation. So within 2 to 3 days of doing electroporation, we like to do an initial pool analysis to see how well the editing took place in our cells. And we take the best one of those 3 and move forward from that point based off the pool analysis that we do. And that's simply done using Sanger sequencing and our proprietary ICE software to analyze those edits. Depending on the type of project the customer is working with, if they want to just stop at the pool, we will then take that and this is where the process diverges. We will then take that pool and do some, let it outgrow a bit to get to the proper cell number that we guarantee our customers that we will provide them, and do that final quality control check on those cells. If they wanted any sort of pluripotency testing on that, we would allow for that to happen at that point. We do some final mycoplasma testing, as well as some final genotyping, just to verify that the cell lines that we are providing are exactly what we said that we would provide to our customers, and then we ship those out. However, if our customer chooses to go down the clonal route and needs a clonal iPSC line, particularly in this instance, this is where it gets fed into that cell nursery that we were talking about earlier. And so we print cells into a 384 well plate, single cells into the 384 well plate, and simply from that point, we just have to allow them to outgrow, to perform the outgrowth, and to get the numbers of cells that we need to provide, that we guarantee that we will provide. Once it gets up to a certain point, we will then re-QC and clonally analyze those cells to verify that we can provide the customer with the specific edit they are looking for. We guarantee at least 2 clones per edit, but we allow up to 5 clones per edit. And so this is where, at this clone analysis stage, where we would pick the 5 best clones to move forward with, and we would continue to let them outgrow until we get to the proper cell number that we need. Then, similar to the pool project, we do that final quality control where we do 1 final round of genotyping. We do all of the karyotyping, pluripotency testing if the customer so chooses, as well as any other quality measures that the customer would like at that point. And then we ship those out and allow the researchers to start their project. So what does this all mean? It means that, for Synthego, it means that we're able to create high quality edits. Looking specifically at some of the types of data that we've recently seen come off our platform, we're able to, particularly at a pool stage, look at edit efficiencies of well over 90% for our in-house workhorse PGP1 cell line, as well as 3 different commercially available cell lines at this point. So really seeing high edit efficiencies. And again, this is at the pool stage for these 4 different cell lines across the board for the particular locus that we're looking to knock out in this project. Not only are we creating highly edited cell lines, we're creating highly functionally edited knockout cell lines. This is confirmed by some functionality testing that we'd originally done with some of the cell lines that were on our platform, in which we knocked out the HPRT gene in a pool of iPSC cells. A bit of background on that: if you knock out HPRT and then expose the cells to 6TG, you would expect the cells to survive. However, if you have not knocked out this gene, you would expect the cells to die. And this can be seen in the images on the right here. We've knocked out the HPRT in a pool of this particular iPSC cell line. And when you look at it, the parental cells that have not had that knockout, you start to see extensive cell death on the upper right panel there. For the cells that have the knockout, you're seeing survival. And then as a control on the bottom, cells in both camps that didn't receive any 6TG, we're seeing the growth that we expect to see based off this. So this confirms we were creating not only a subpool that is highly knocked out with a highly knocked out gene editing efficiency, but we're also seeing highly functional knockouts of these iPSC cell pools. Again, where we're probably leading down the path today is creating these, particularly for disease modeling, but creating precise SNPs or SNBs in iPSC cells. This is a bit of a report that we send out to customers and something we've become quite well at doing. But in this particular edit, we've been able to cleanly, with 100% success rate, convert the C to a T in this particular cell line. And this really allows you to create, and this is a clonal cell line, and really create a highly accurate disease model for whatever you're looking to do in your lab. SNPs are a bit easier, but what about small tags? So again, this can be a little bit more difficult depending on the size of the tag, but this is something that we've become really well and adapted doing on our platform based off of our automated skill sets and things like that we've been able to create. So we've used our workhorse PGP1 cell line in this instance and looked at knocking in various tags, 6 or 7 different tags into a particular locus to look at our efficiencies with this. When you look at our knock-in scores across any of these tags, these various tags from HA to a 3X flag tag to a single flag tag to V5 to a MIC tag, we see really high knock-in scores in a pool population. This can really be seen with the HA tag when you look at the fluorescent staining there and overlay that with the DAPI staining. It looks like essentially, every cell has been tagged with the HA tag. When you look at the pluripotency marker here, the SSEA4, we see that these cells are still highly expressing the pluripotency marker, and they haven't begun to be differentiated, which is really exciting to see. In fact, we don't use any enrichment or selection on this. And if you look at this from a pure Sanger sequencing standpoint, these HA tag iPSC cells that are in pools are actually looking very clonal-like, right? And essentially, we're getting close to 100% knock-in in the pull stage with this HA tag. I think on here it says 98%. So that's a pretty good knock-in efficiency at the pull stage for a small tag. This, is again representative images of the that we saw on the other screen there with the HA tag fluorescence image in the SSEA4. Again, we know it's important that we maintain differentiation on our platform. Gene editing can be stressful, or maintain undifferentiated on our platform, cells on our platform. We know that gene editing can be stressful in cells, and so we used to perform a standard some fluorescence imaging, but we never saw the cells that we were put in the platform to come on the back end to undergo any sort of differentiation. And so this is just some of the images of some of the stuff that we used to get out on the back end when you look at NANOG, trial 181, OCT4, trial 160, SSCA4, and SOX2. As you can see in the fluorescence imaging there, the cells that we get on the back end of the platform are still expressing all of the pluripotency markers that you would expect them to express. Again, just another slide highlighting the types of add-ons that we allow our customers to add on to any project, which is this pluripotency testing that we do through PluriTest, and karyotyping testing that we allow. Obviously genomic stability is important as well, and we can measure this as well and provide this on the front end and the back end of any project. So what do we deliver to our customers in the form of iPSC projects? It really depends on the format, but from a pull format, we provide 2 vials of either the gene edit of choice, whether a knockout or a knock-in, and 500,000 cells per vial. We also provide, as a standard, as a control, 2 vials of a mock transfected cell pool, which simply consists of the cells that have been transfected with the Cas9 protein only and no guides. So it's just a Cas9 transfection as a control. From a clone format, as I said earlier, we provide 2 vials of 2 independent clones. We allow up to 5 vials, so that 3 additional clones to be purchased. But those come in the form of 500,000 cells per clone per vial on that as well. And at the same time, we provide 2 vials of the mock transfected pool with the Cas9 only. We do have, as I said earlier, the optional add-ons for iPSC only projects, which is pluripotency testing and karyotyping analysis. Up on the right there, you see an example of a certificate of analysis that we provide, which includes the passage number of cells per vial, the results from either the optional testing as well as the mycoplasma testing. So how does this get into CRISPR engineering for cells that enable disease modeling? I think this really comes back to to the Food and Drug Omnibus Reform Act of 2022. I think when this came around, it really allowed drug manufacturers to use any sort of non-animal method, such as cell-based assays or organs on a chip, or even computer modeling for assessing drug safety. And I think this is really where we see the importance of creating these high-quality and highly efficient cell-based model systems that we've become really apt at being able to do. And it really shows the importance of them, because why would researchers continue to use any sort of animal-based model when we can create a cell-based model via gene editing, and then some of the downstream types of assays that the BrainXell can do from a differentiation standpoint. So what does this mean? I think we've talked about, particularly at the Synthego, I think we talked about earlier, we created this highly automation-driven parallel process platform that allows researchers to study multiple genes at one time and multiple edits at one time, resulting in these quick turnaround times so they can quickly analyze their results. This is really, obviously on our platform, fueled by our Halo technology, which is our guide, our synthetic guide platform that gives us really great reagents to start these projects with. And our expertise is, as I'm able to say, as you've seen with even our transfection optimization protocols and things like that, really allows us to establish this rapid testing of conditions to provide the best and highest qualities of edits to our customers whenever they come to us. I think this can really be seen in the research that was done for the Alzheimer's and related dementias, particularly in the Indy project. So as everybody's probably aware of, but may not be well aware of, greater than 6 million people in the U.S. are living with Alzheimer's, right? we know that 1 in 3 seniors will die with Alzheimer's or some sort of related dementia. And well over 60% of those Alzheimer's patients are women. Due to some of the great research that's already been done, we know that there are some 50 genes or approximately 50 genes that have been identified with Alzheimer's and the related dementia, but we don't know how these genes interact and interplay with each other and what particularly is causing Alzheimer's and these related dementias to play out like that, depending on the genes that are affected. So it's really key to identify the linkages between these different genes and understand how they're working together. And the only way to do that is by producing mass numbers of, particularly in a cell-based system, mass numbers of cell-based models to replicate the types of mutations that you see in these patients. Where Synthego really came into play here is our work that we did with the NIH as a part of the INDY project. The goal of the INDY project was simply to generate a standard collection of quite literally hundreds of iPSC cells that carried many of the common ADRD relevant mutations that could be provided to researchers around the globe to help expedite and move along the research that was done in AD-RD. But as we know, and as we've talked about, there's many challenges that were associated with that. Obviously, CRISPR is hard. Not only do you need high quality chemically modified reagents, but as we talked about earlier, not every cell line is the same, not every condition is the same, and so creating optimized conditions to get the highest quality edits can be difficult. Oftentimes, this leads to a low success rate. From a benchmark report that we did several years ago, we found that researchers often failed 7 times before they were able to achieve a successful edit. And now what that means is that if you want to scale across multiple genes and multiple cell lines with multiple edits, that's going to take not only time, but tons of resources that many researchers just do not have. So what was Synthego able to do in this instance? In a matter of 5 months, due to our highly automated platform, we were able to generate 264 individual clones for researchers to be able to move forward Alzheimer's research. What this accounted for was 22 independent gene targets and 12 variants per gene target for those 264 clones, or 264 clones that we generated. Take into account, we did this in less than 5 months, or right around 5 months. I think this really goes to show the power of this automated platform that we were able to be able to allow researchers to move forward quickly with being able to move their research forward very quickly and identify cures and treatments for many of these diseases. I think a lot of this can be summarized by what Dr. Scarn says there down at the bottom. He says offering CRISPR edits at scale with Synthego, which we were able to do at Synthego, similar to what we did with our single guide reagents back in the day, puts researchers on the cusp of being able to study thousands of genes and examine hundreds of variants of those genes, as we saw with the 264 that we were able to generate. This allows scientists to move more faithfully, or to more faithfully model the complexity of human disease, which could lead to development of therapeutics and drugs or next generation therapies for many serious diseases. And I think we can do that at a very fast pace now due to the highly automated platform that we have. So as I wrap up here, and before I pass it over to the BrainXell team to talk about some of the downstream assays that can be done with these edited iPSC cells, I hope that I've been able to kind of get across a lot of what's important to us here at Synthego, which is taking engineering approaches, taking these machine learning and automation, and really applying that to biology, to where we were able to have predictive CRISPR designs, really optimized transfections to work with any cell line that the customer would want to work with, and really been able to drive research across the whole drug discovery platform, from early research phases in the more or less cell lines downstream to the iPSC cells that move more and more closer to the clinic with these iPS cells. I will finish up before I pass off to Seth and say we do have 6 cell lines in which we've completely optimized that are Synthego supplied from an iPSC perspective on our platform that we are really well at working with. One I talked about earlier, which is that PGP1 cell line, which is a bit of a workhorse cell line for us. But of note, we have now partnered with BrainXell and have 2 to 3 of their cell lines on our platform now and which we're able to start projects with right away to create these highly functional either knockouts or knock-ins for our customers to start the research right away. So with that, a big thank you to everybody for giving me their attention, and yeah, thank you.
All right, thank you, Travis. I am excited to talk about BrainXell today and talk about how we can partner with Synthego and how we can take those cells that have been edited by Synthego and turn those into fully functional neurons, microglia, and astrocytes, and allow researchers to do some really interesting work. So BrainXell is a company that specifically focuses on the stem cell to differentiation of neurons. And we really fit into this world where we're trying to bring the most relevant neuronal and glial cell models to researchers. And that includes basically taking the human cells and turning those into human neurons that can be used for all sorts of different research reasons, but one of the big reasons is drug screening. So taking those neurons and seeing how they can be impacted and affected in a drug model. So at BrainXell, we have a very tight focus. We work on cells of the human central nervous system. We have a very strong expertise within this field. This is the only thing we work on is neuro, and we really view it as we want to create the highest product quality and performance in the researcher's hands. So that means we're not happy until the customer is happy. We also do a lot of custom production. So we have our cell lines that are available, as Travis mentioned. We've got several different cell lines, and those are control lines. But we can also work on customer provided cells, whether they are edited or patient lines that have been corrected. And so we really try to support each customer project before, during, and after delivery of those cells. So why do we focus on neuro specifically? Neurological disorders is a pretty devastating experience. In 2019, there was over 850,000 deaths worldwide. But really, more to the point, there are millions and millions of people that are living with disorders, things like Parkinson's disease or Alzheimer's disease, multiple sclerosis, migraines, epilepsy. A lot of these things don't necessarily kill somebody, right away, but they can be things that people live with for many, many, many years. And so this is a really significant area for research. And if we look at dementia alone, 1 in 10 people, over the age of 65 have dementia. And we see that this is actually more prevalent in women than in men. There's also a lot of rare disorders. So disorders that don't have a huge, patient population, but there's a tremendous need there. And so why human neurons? There are a very high rate of clinical trial failures. Neurology specifically has had a very low success rate in terms of drug approvals. That 5.9% is really, really low when it comes to other cell types. And efficacy is really the main cause of drug failures. It's not always safety, it's not always strategic or whether the dollars are there, but these things just fail because they get late into clinical trials and there's just not proven efficacy. And a lot of this is because of lack of understanding of the disease, a lot of target validation that hasn't been done properly, and in vitro cell culture is used for the screening of these potential therapies. If you're screening in animal models and you're looking for hit validation in animals, you may be setting yourself up for failure. So what do drug companies do? What do people do that are doing drug discovery? There's a lot of in vivo work. This is always done in rodent or non-human primates. And it's a very high cost, low throughput, and high variability endeavor. And then there's really that questionable translatability. If you're working in a mouse and you're providing a drug and you're trying to create a therapy for humans, how effective is that? Is that drug going to actually be? It's often an unknown. There's also in vitro models. Traditionally, people in drug discovery have worked with rodent lines, whether it's primary mouse or primary rat cultures, or cancer lines, glioblastomas. The problem with those is, they're not always human, and they're not always true cells that you want to actually target for the therapy. So with human iPSCs, this gives us the ability to generate these different neuro subtypes that are really specific for different diseases. So many diseases have a very specific type of neuron or region of the brain that is targeted. In ALS, we're looking at motor neurons. In Alzheimer's, you want to work with specifically cholinergic neurons. In multiple sclerosis, oligodendrocytes. Parkinson's, it's dopaminergic neurons. And so if we look at all these different disorders, there are different types of cells that are really valuable to study specifically. And if you're working in that primary rodent culture, you can often get a mixed population of things. And so you don't necessarily know, is your drug affecting one cell type or another and causing an effect? And so that's where we come in, is to be able to produce these very, very specific cell types and make those available for that drug testing. And so, how do you work with this? You want to get disease line mutations. So there are often known mutations that increase the risk for a disorder. And you can get these, you can source these from either patient lines. So a patient who has a disease has donated their cells to a biobank, and those cells have been reprogrammed into iPSCs. Those iPSCs can then be differentiated into neurons. You can also start with a non-disease line, so a control line, and do that same differentiation. But you can also start with the patient line and use the CRISPR-Cas9 system to correct that mutation. And so now what you can have is a genetically corrected iPSC that can be differentiated into a neuron. And so now you have these control neurons. And then you can also start with the patient model, and you have the disease neurons. So now you have the disease line and the isogenic line, and you can start to do a study to compare the two. The other option is starting with a control line and generating that specific mutation, in the control line, and now you have mutation in the control line, and the control line as a healthy control. And finally, what do you do with these cells? These cells are going to go into a variety of different assays. You can do functional assays where you're looking at electrical activity. You may do some ICC on the cells and look at the actual health of the culture. But then you can also do a lot of transcriptional analysis, where you're going to grow the healthy neurons and the disease neurons side by side and look at the transcriptional output, look at pathways that have been upregulated or downregulated. And maybe you're going to try to start finding pathways that can be exploited with a variety of different means, small molecule, large molecule, to try to make that disease neuron more like the healthy neuron. And ultimately, we want to be able to create treatments, right? So the hope is that we can understand these pathways a little more clearly, and then from there create treatments that can help people. So one of the projects that I want to highlight here is revolving around TREM2 and ApoE. These are two genes that are found to be involved in Alzheimer's, and in recent years, they have become much more in the interest to researchers. So what is TREM2? TREM2 is a gene that is transmembrane protein that sits here and will start to basically bind onto a lot of different items. These will then get phagocytosed and create pathways that lead to all sorts of different things, right? So TREM2 is predominantly expressed in microglia. So microglia have a lot of TREM2 sitting on their cell surface, and they're out there gobbling up things like lipids and amyloid beta and myelin that has been broken down and apolipoproteins. This then triggers various pathways that lead to inflammation, proliferation, survival, migration, and can impact not just the microglia, but the neurons that are surrounding them. And a loss of function of this TREM2 really leads to a failed immune response. And this can then trigger inflammation. It can trigger all sorts of things that ultimately can create a risk for Alzheimer's disease. Now, if we look at TREM2 specifically, there are different Alzheimer's disease associated variants. R47, R62H, D87N, T96K are all known to be risk factors for Alzheimer's disease. And so what we can do then is create an iPSC line that has these different mutations and turn those iPSCs into microglia. The same thing can be said for ApoE. ApoE is a apolipoprotein that is involved in lipid and cholesterol transport, right? So we've got the ApoE, and it is going to basically process these things like cholesterol and phospholipids, and the astrocytes and microglia are going to gobble that stuff up. If we have a specific variant of ApoE, we have found that E3, so this ApoE3 is at two sites, cysteine and arginine. This is kind of the wild type or control ApoE3. This is basically just standard Alzheimer's disease situation, but an E2 variant where the one of the cysteines is intact and the other arginine has been converted into a cysteine. This is actually found to be protective against Alzheimer's disease. Whereas the E4 variant, that cysteine is turned into an arginine. And so now we have an arginine and another arginine at these two sites. This is known to be at increased risk for Alzheimer's disease. Okay, so what did we do? We worked with Synthego to generate several different edits. We have our BX32 iPSC line. This is this comes from an Asian female, age 29, with no known neurological disorders and a known ApoE E3E3 genotype. So we took that line and generated the E4E4 mutation. And then we also took that original line and created a TREM2 knockout. So in doing that TREM2 knockout, we are causing a disruption right about here in TREM2. And that's going to cause a basically a nonsense frameshift mutation. And then that TREM2 protein is no longer functional. We also created the TREM2 R47H. So this is an intact TREM2 protein, but has just that SNP modified. We've also created a TREM2 knockout with the E4E4 mutation and a TREM2 R47 with the E4E4 mutation. So now we've taken that one control line and generated 5 new additional iPSC lines. And this allows us to create a whole bunch of different testing scenarios where you can compare many different genotypes and compare them all back to that isogenic control. So we took, this is a long project, but we're starting with the TREM2 knockout and the TREM2 R47H, differentiating those into microglia. With the microglia, we can look at the morphological similarities and differences. And then we can also look at different expression patterns. TMEM119 and P2RY12 are both microglia markers. So we can look at, are these truly microglia? And yes, they are. And even though they are true microglia, we do see some significant changes morphologically and phenotypically. And then we want to start looking at some functional assays. So because microglia are involved in phagocytosis of beta amyloid, we've acquired a beta amyloid that has a fluorescent tag on it, in this case, HL488. So what we can see is as the microglia are in the presence of that fluorescent beta amyloid beta, we can actually start to see the beta amyloid enter the microglia as they basically chew up the A beta. And we can compare that phagocytosis over the multiple different mutations. So we can compare wild type to a TREM2 knockout, compared to a TREM2 R47H, and compare it to an ApoE4E4 mutation. And what we found was that the TREM2 knockout really severely impacted the beta amyloid uptake. So at day 9 in culture, the TREM2 knockout has significantly reduced ability to uptake that beta amyloid. If we look at the TREM2 R47H, it is slightly affected, but not nearly as severe as the knockout, which actually makes sense. And then the ApoE4E4 doesn't appear to be to be affected if we look at the percentage of cells that have taken up that amyloid beta. However, it's not just about the number of cells that have taken it up, but the intensity of the A beta that they've taken up, right? So if one cell takes up just a fraction of A beta compared to another cell, we can really track that as well. And so we found that if we look at the mean fluorescence per cell, we do see actually an impact in the ApoE4E4. So there is something that is reducing the ability for that microglia to uptake the A beta, even in that ApoE4E4. And so I think this really kind of emphasizes the importance of looking at multiple different perspectives. When you create this edit, you don't want to just look for one single thing. You want to look at a couple different ways to measure. And what do we want to do next? We really want to start to add in different components. So we can start to mix and match the microglia with neurons and glia. So astrocytes and glutamatergic neurons, astrocytes that have the exact same mutation and glutamatergic neurons that are wild type, or vice versa. Really start to build out some interesting models of GLUT neurons and astrocytes and microglia with all these different mutations. So there's a lot of potential work that researchers can do. We can also start with patient lines. So if we start with a patient line and maybe do an edit, that's another approach. So here we have a publication that just came out, and this is a group that is working in Dravet syndrome, and provided a patient line to us to differentiate into GABAergic neurons. And so, starting with that patient, the iPSCs are generated and then differentiated into GABAergic neurons, and then plated on high-density microelectrode arrays. So we can look at those GABAergic neurons and find that they are really expressing the MAP2 neuronal marker and also the GABAergic markers. But we can also look at the specific mutation that is involved in that syndrome. So in this case, SCN1A, and look at the changes of that over time in qPCR and in RNA-seq. So this D through G is looking at single cell RNA-seq and comparing the different components. So those lines can then be from the GABAergic neurons can be plated on, in this case, high density microelectrode array, a Maxwell system, and start to look at different components, right? So you're looking at percent of channels that are active. You're looking at firing rate. You can look at amplitude and look at this over time. So here we're seeing day 14 in culture all the way out to 42 days in culture. And you can start to see differences. What you also can do is start to apply different drug treatments. So in this case, take those GABAergic neurons that are derived from a patient and at day 42, apply a drug. So apply PBS, which is going to be your control, or a couple different treatments that, so the muscimol is a GABA receptor agonist, and that's going to be looking for functional GABA signaling. And then HM1A at different concentrations. This is a NAV1.1 potentiator. And so we can look at how this impacts that electrical activity as well. And the goal here is to create a couple test compounds, and then also maybe your drug treatment, and look at how does your drug treatment impact this firing rate in comparison to the disease line. Well, one of the big things here was that a limitation of this study is the lack of an isogenic control. And so that's where the CRISPR editing can come in to take that patient line, that SCN1A mutation, and make that correction. And now we can generate all this data in a control line and the disease line, and test different test compounds and really start to get a really nice, interesting way of screening, whether it be high throughput or medium throughput, to find some novel drugs that can alleviate these disorders. And with that, I will thank you for joining me and Travis in talking about the this CRISPR editing, and then also the complementary differentiation into human neurons.
Excellent. Well, thanks so much, Seth and Travis, for a great presentation. And I'll just invite Travis and Seth back online for the Q&A, and we can get started here. All right. So first question, and Travis, I think this one will be for you. So you presented a lot of detail about your capabilities to edit the iPS cells, but do you work also with primary or differentiated cells like cardiomyocytes or hepatocytes?
Yeah, that's a great question. Currently, we don't offer a product in the primary cell type. We're looking to develop other type of editing capabilities. One of the things we're looking to launch probably in the very near future is obviously primary T cells. So looking in those immune cell types and doing editing and those types of cells. We have done some dev work in cardiomyocytes, previously, and we're looking at maybe possibly developing those in the future. So some new areas coming, hopefully, but nothing specifically that is offered right now.
Excellent. Thanks a lot. Seth, I think this question is for you. So how long does a project take, and for CRISPR and for the neurons?
Yeah, I mean, it can really kind of depend on the cell type. and also the scale, and also the iPSCs. The iPSCs have a big role to play in terms of being able to differentiate. Most of our projects, we say, will take about 4 to 6 months to create, from the time we receive iPSCs to the time we're delivering the fully differentiated neurons or glia cells.
And maybe I can tag team on that a bit, Seth. If you look at the CRISPR side of it, from our knock-in perspective of clones and iPSCs, we're looking at 10 to 12 weeks on average turnaround time for us to provide this to Seth, if it's a sort of a combination project of here's introducing the edit and then providing it to the BioCell team or the brain cell team to do the differentiation downstream.
Yeah, and I would say it probably it's a little bit faster or less delay if it's using one of the Synthego lines that are already banked.
Correct.
Right? That's why we have ours with Synthego already.
That is true. Yeah, we already have those on. We've validated those on our platform and can kick those off right away and know how to work really well with them at this point.
Mm-hmm.
Awesome. Yeah, thanks both for your input. All right. Do you offer any reprogramming services?
I can take that from our end. We don't, but this is why we have a great partner, I think, in BrainXell. I think, Seth, you can talk about this. I think you guys do some of this. We've reached out to you before for offering some of these.
Yeah.
We don't.
Yep. So BrainXell, we do reprogramming as well, primarily from fibroblasts. We do have the capability to do other forms, but fibroblasts using mRNA reprogramming is our primary approach. We are really a stem cell company that, that's what we focus on is working on the stem cells. So whether it's coming from fibroblasts, turning those into stem cells, and then turning those stem cells into different types of neurons.
Fantastic. I think in the interest of time, we'll just make this next question the last one. So forward-looking question. So are there any upcoming developments or new research avenues coming out that you're particularly looking forward to? Travis, maybe you could start.
Yeah, I can start there. As I mentioned earlier, we're really excited. We're getting into editing, primary immune cells, CD4 positive T cells, CD8 positive T cells. Really driving that forward, and that should be upcoming in the very near future. From outside of iPSCs, if you look at just a more light standpoint, where our express cell offering is really something we're driving forward. So we're getting pretty close to being able to turn around a pool of edited cells in about 7 days, 14 days at this point. So really something we're excited for our platform. Seth, I don't know what you have.
Yeah, from our perspective, neuro is all we do, and so we're always looking to develop new neuro subtypes. So we have a couple different types of cells that are in the works that we should be releasing probably mid-year this year. It's always nice to kind of add different types of cells that are really relevant for different disease types. So that what's really cool is if you edit an iPSC, you have that to then differentiate into multiple different neural subtypes. So you can generate glutamatergic neurons today, and in the future, if a new subtype comes out, oligodendrocytes or sensory neurons, those are always something that can be created as well from that same line.
Excellent. Yeah. Lots of work to do, but definitely a lot to look forward to as well. Maybe we could just squeeze one more question in there. I think it's a good one. Are there any IP or commercialization restrictions if we use the BrainXell iPSC lines?
No. So the cell lines that we have are wholly owned by BrainXell, and we provide them for research. Our philosophy is once they're produced and are out there for you to use as research, they become yours. So that's the benefit of working with our lines. So
Awesome. Great to hear it. All right. Well, thanks so much, Travis and Seth, for all the great insights today. It's really been a pleasure having you with us.
Awesome. Thank you, guys. Thanks, everyone, for joining us.
Yeah. Thanks, everyone, for joining.
Yeah, definitely. And in closing, we hope you enjoyed the Scientist.com, Synthego and BrainXell webinar, and we'll see you again next time. Have a great day, everyone.